Hello everybody, my name is Evelyn Volke and I'm a professor at Ghent University. Let's talk about the process operation and control of aerobic granular sludge reactors. We will start with an introduction on aerobic granular sludge and aerobic granular sludge reactors. We will see that avoiding diffusion limitation is the most important prerequisite for growing granules. We will study the batch-wise reactor operation and the corresponding concentration profiles in the lab as well as for full-scale plants. Then we will discuss some specific process control aspects associated with the batch-wise operation. And we will end with some additional selection pressures which can be applied to obtain granular sludge. Let's start by having a look on what is aerobic granular sludge and what are aerobic granular sludge reactors. Aerobic granular sludge is defined as aggregates of microbial origin which do not coagulate under reduced hydrodynamic shear and which settle significantly faster than activated sludge flocks. Typically, aerobic granules are larger than 0.2 millimeters. However, the fact that the granules do not coagulate is more important than the size criterion. The sludge volume index, or SVI, is the volume occupied by 1 gram of sludge after settling for a certain time. For aerobic granules, the volume taken up after 5 minutes of settling is about the same as the volume which would be taken up after 30 minutes of settling. So the SVI 5 is about the same as the SVI 30. This is a scientific way of expressing that granules settle very well. Another important feature of aerobic granular sludge is that there is no compression of the settled sludge, unlike for activated sludge. Okay, so we know what is aerobic granular sludge. Let's see what an aerobic granular sludge reactor looks like and how it relates to a conventional wastewater treatment plant. In a conventional wastewater treatment plant, the removal of carbon, nitrogen and phosphorus components takes place in different reactor compartments. These compartments take up a lot of space. Besides, wastewater needs to be pumped around between these compartments, which brings along pumping costs. Another problem in conventional wastewater treatment is that the bacteria which purify the water grow in flocks. The density of these flocks is only slightly larger than the one of wastewater, which makes that their separation process proceeds very slowly and requires large settling tanks. This picture shows water with sludge flocks after 5 minutes of settling time. In the case of aerobic granular sludge, the bacteria grow in granules. The removal of carbon, nitrogen and phosphorus components takes place in different zones of a single granule, so there is no need to pump around the wastewater between different compartments. The granules are closely packed in the reactor, which results in a most compact installation. The granules settle very well, and they can thus be separated very easily from the purified effluent. This is done in the reactor itself, so there is no need for settling tanks, and that makes the installation even more compact. Overall, aerobic granular sludge reactors take up 75% less space than activated sludge plants. They consume up to 50% less energy and require 30% less investment costs. But how to grow granules? Well, as was already made clear to you in the lecture of Mark van Loosdrecht, the most important prerequisite is to make sure that diffusion is not limiting. Indeed, if we look at how bacteria organize themselves, we see that in case of diffusion limitation, bulking sludge is obtained. This is the case when the substrate concentration is low or the biomass growth rate is high. The substrate is limiting, and the bacteria reach out in all directions to get food, resulting in filamentous structures. At the other side of the spectrum, there is granular sludge, which is obtained when there is no diffusion limitation. This is when the substrate concentration is high or the biomass growth rate is low. The substrate is not limiting in this case, but the bacterial growth as such is the limiting factor. This results in the growth of bacteria in the form of dense granules. And in between bulking sludge and granular sludge, there is a flocculated sludge, as is present in typical activated sludge wastewater treatment plants. Okay, so now we know that diffusion should not be limiting, which means that the substrate concentration should be relatively high or the growth rate should be relatively low. This is relatively easy for slow-growing microorganisms, as is the case for microorganisms involved in anaerobic digestion processes. In that case, one can even grow granules for high concentrations of soluble substrate, as is done for instance in a USB reactor. But how can we possibly grow granules of heterotrophic bacteria, which convert organic carbon, and which are known to grow very fast? The trick is to minimize substrate limitation, which in this case means to minimize the limitation of organic carbon. Also, oxygen should not be limiting. So now we know what to do. We need to minimize substrate limitation for heterotrophic bacteria. But how is this done in an aerobic granular sludge reactor? Well, we will have an anaerobic feeding phase. Given that the feeding phase is anaerobic, there will be no aerobic growth of heterotrophic bacteria, which we know takes place very fast. The biodegradable organic carbon, or BOD, will be taken up by PAO and GAO, phosphorus accumulating organisms and glycogen accumulating organisms. They will use it to produce storage polymers. Next comes an aerobic phase. This is a famine phase, given that there is no carbon left for other bacteria than the PAO and GAO. So the PAO and GAO grow on intracellular storage polymers, which is a slow growth and not diffusion limiting, not diffusion limited. So in summary, aerobic granules are grown by making sure that diffusion is not limiting. This is done by separating the substrate uptake and the biomass growth, by having a separate anaerobic feeding phase. The bacteria grow on internal storage polymers. They are slow growers, and the anaerobic feeding phase selects for these slow growing organisms, which results in the formation of dense, stable and smooth granules. Let's now zoom in into the batch-wise reactor operation. Aerobic granular sludge reactors are operated in sequencing batch mode. In the lab, it looks like this. We start with the 1 hour feeding phase through the settled granular sludge bed. Like this, the biomass sees a high substrate concentration. The organic carbon substrate is converted into PHA, a storage polymer. Since there is no aeration, there will be no aerobic growth. At the same time, the PAO are releasing phosphate. And here are the corresponding concentration profiles. Ammonium accumulates as it is fed and not converted. There is no aeration. Organic carbon is rapidly consumed and converted into storage polymers. At the same time, phosphate is released. And some nitrate, which is left over from the previous cycle, is converted. Once the reactor has been filled with influent, the 2-hour aeration phase starts. Now oxygen is available for the aerobic microorganisms to grow. Given that there is little to no substrate left in the liquid, only those bacteria that have stored the substrate as storage polymers can grow. Ammonium will be nitrified to nitrite and nitrate by nitrifiers which are present at the outside of the granule. Subsequently, nitrite and nitrate are available for denitrification. Denitrification takes place using storage polymers as organic carbon source. Remember that the organic carbon from the influent has been completely converted. Denitrification is performed by bacteria which are located at the outside of the granule, typically PAL. The influent phosphate and the phosphate which has been released in the anaerobic phase will now be taken up and stored as polyphosphate. And here are the corresponding concentration profiles. Ammonium is nitrified to nitrate, and phosphate is taken up. After the aeration phase comes a settling phase, followed by effluent discharge. Rocks and small particles that do not settle fast enough will be washed out. Fast settling biomass, like granules, will be retained in the system. And here you have the concentration profiles all together. Inside the granule, it looks like this. There are the nitrifiers in the outer layers and the denitrifying phosphorus accumulating bacteria at the inside. Let's now move on to full scale. Full-scale aerobic granular sludge reactors are also operated in sequencing batch mode, with a fill and draw phase, an aeration phase, and a short settling phase. But the main difference with the lab-scale operation that I just presented is that the influent feeding and the effluent withdrawal take place simultaneously. Simultaneous influent feeding and effluent discharge has several advantages. It saves time and energy, and it makes that the discharge flow can be kept relatively low. And here you have the concentration profiles during a typical day of operation in a full-scale granular sludge installation. Let's isolate one cycle, which takes about 6 hours. And now stretch the profile a bit so we can see it better. The orange line indicates when influent is fed. The blue line shows the oxygen concentration profiles in the reactor. We see how nitrate is consumed during anaerobic feeding and produced during the aerobic reaction phase. This is because during the aerobic phase, nitrification takes place, so ammonium is converted to nitrate. Phosphate is released during anaerobic feeding and taken up during the aeration phase. Overall, the dynamic concentration profiles which are obtained during batch-wise operation contain a lot of information. The slope of the concentration profiles is related to the biological conversion rates and to the volume exchange ratio, that is, the fraction of the reactor volume which is replaced with fresh wastewater during each cycle. All of this information is very useful for process monitoring and control. This batch-wise operation also results in a good effluent quality. In a continuous stirred tank reactor, the concentrations in the reactor are equal to the effluent concentrations, which are typically required to be relatively low. Operating the reactor at such low concentrations may lead to substrate limitation and thus to low bioconversion rates. During batch operation, the substrate concentrations in the reactor decrease over time. From the influent concentration at time 0, which is typically high, to the required effluent concentration at the end of operation. As a result, the substrate concentration in the reactor is high most of the time and will not be limiting the bioconversion rate. The effluent concentrations can thus become very low, without lowering the overall conversion rates. The batch-wise reactor operation also involves specific process control aspects. First of all, why do we need process control? Well, we need to meet the effluent criteria for organic carbon, expressed in COD, for nitrogen and phosphorus. We want to avoid suspended solids in the effluent. We want to optimize the sludge retention time, or SRT. And we need to manage all of this dealing with variations in influent flow rate and the influent composition. The next question is, how can we control the process? What are the control handles that we have to influence the process? As already indicated, the batch-wise dynamic reactor operation makes it possible to monitor the biological conversion rates over time. Furthermore, set points can be adjusted, cycle and phase length can be controlled, and can even be adapted according to the influent conditions. The last option is called batch scheduling. You will learn more about that in the lecture of Edward van Dijk. In this lecture, we will have a look at more basic control aspects of the sequencing batch reactor during the fill and draw phase and during the aeration reaction phase. The fill and draw phase is characterized by anaerobic conditions. The influent is fed from the bottom through the settled sludge bed. Organic carbon is taken up and stored, and there is no aerobic growth. The length of the fill and draw phase is typically 1 hour. It needs to be long enough to ensure the uptake of readily degradable organic carbon. This will depend on the influent COD phosphorus ratio and on its BOD-COD ratio. There is also a limitation regarding the volume exchange ratio, or VER, which is the ratio between the volume of the fresh influent, which is fed in a cycle, over the reactor volume. The volume exchange ratio always needs to be lower than 65% to make sure that the influent wastewater is not mixed with the clean effluent and to prevent sludge washout. Finally, there is also a requirement for the upflow velocity in the reactor. The upflow velocity is calculated as the influent flow rate divided by the reactor cross-section area. The upflow velocity must remain lower than 5 meters per hour to prevent sludge washout. Next comes the aeration phase. In this phase, there is almost no readily biodegradable organic carbon left in the liquid. There is slow growth on intracellular storage polymers and simultaneous removal of organic carbon, nitrogen and phosphorus, as we have discussed earlier. The length of the aeration phase is controlled in order to allow nitrification and phosphorus removal. In some cases, it is chosen to apply chemical phosphorus removal besides biological phosphorus removal. The oxygen set point will determine how deep oxygen will penetrate in the granules, and in this way also the extent of simultaneous nitrification-denitrification. In some cases, there is also an additional phase with on-off aeration. This is done when extra denitrification is required. And let's now have a look what are possible additional granule selection pressures that we can apply to grow granules in the reactor. We already know that the prime prerequisite for growing granules is to make sure that diffusion is not limiting. This condition is fulfilled by having an anaerobic feeding phase for substrate uptake and an aerobic reaction phase for biomass growth. This is the most important condition, but it is not the only one. Another important selection pressure is selective wasting. Selective wasting is based on the observation that the reactor always contains some flocculent material, besides granules. The flocculent material consists of inert particulate organic carbon, biomass which is grown on organic material that remains after anaerobic feeding, and of eroded material from the granules. In selective wasting, the top of the sludge bed is removed after a short settling period. As a result, the flocculent biomass fraction in the reactor will be retained in the reactor for less than 5 days, while the granules will have an SRT of about 30 days. So selective wasting selects for granules based on their good settling properties. And how about shear as a selection pressure? You see that I put shear between brackets, which already indicates that it will not be a main selection pressure. In the literature, there are definitely indications that applying shear will help to create smooth biofilms or granules. However, it requires a lot of energy to create shear stress on the granule surface, and also it is not needed as long as you stick to the first requirement to grow granules, which is to make sure that diffusion is not limiting, and which you can do by separating substrate uptake and biomass growth. Besides avoiding diffusion limitation and besides applying selective wasting, an additional selection pressure to grow granules is plug flow feeding. In aerobic granular sludge reactors, the influent is fed from the bottom of the reactor through the settled granular sludge bed. In this way, diffusion limitation is minimized and the substrate can be taken up over the whole depth of the granule. Moreover, in this way, the larger granules at the bottom of the reactor receive the most food, while the slow settling flocks at the top receive less food. So there is a type of self-stabilizing effect. So, that were the most important tricks to grow granules. However, there are even more selection pressures which can be applied. For more information on that, I refer you to the lecture of Edward van Dijk. That's all for me today. Enjoy the rest of the course.